My name is Hyun. I'm a postdoc in Lord Frank's lab at UCSF, and today I wanted to share with you how our lab has been adopting NWB by placing it in the context of a scientific data pipeline. So I'd like to start by noting that NWB is a great tool with many nice features. It is a standardized format for the neuroscience community that enables easy sharing of data. It sort of naturally provides the metadata along with the data so that you can make sense of what other people's data set is like and actually reuse it for the particular questions that you're interested in. It also comes with support from a team of developers who not only maintain the format, but also educates the community in a workshop like this one. And finally, it is supported by and recognized by an ecosystem of other data analysis tools that solve each solve specific problems. And I think this is really sort of where the benefit of doing science with NWB becomes evident. My talk will basically try to illustrate that by showing how we placed NWB in a data pipeline. So by data pipeline, I just mean basically a set of steps that takes us from the raw data to scientific conclusions. And of course, this includes a lot of steps. We've already heard some of them, such as data cleanup, aligning the data with other data streams like behavior and neurophysiology data, spec sorting, and visualization and model fitting and so on. Many of which we would like to standardize because and automate to the extent that they're possible, because we don't all want to be solving this problem over and over again. So we basically started by asking ourselves what it is that we want from such a pipeline. And one thing is that we want it to be easy to use. So by that, I mean, the user would be insulated from the nitty-gritty details of how every step in the process was implemented, although they can find out about them if they wanted to. It should also enable consistently reproducible analysis and also minimize the effort that is required for sharing the data and the analysis code. And finally, support team science at all levels. By that, I mean collaborations within the lab as well as across labs. And thanks to the effort by many people, we now have tools that can solve, that if combined correctly, can solve many of these issues. So Neurodata Without Borders, obviously don't have to talk about that, but you may also have heard of DataJoint, which is a tool that allows you to basically create, maintain, and manage a relational database using Python or MATLAB for scientific data. And I think there was a talk earlier about Dandy, which is a public repository for neurophysiology data. But the question still remains as to how an individual lab can best combine these tools in a productive way. After all, these tools have some degree of complexity associated with them. And as a result, the user has to spend quite a bit of time and effort learning about them and thinking about, what is the best way to combine these tools for their own needs. So today, well, for the rest of the talk, I'd like to sort of describe to you the solution that our lab has converged on. So we start with the raw data, obviously, and because we do experiments in rats, we record from rats as they navigate in complex environments. We have multiple data streams. So we have electrical recordings from tetras and probes, also auxiliary digital input-output signals that tell us information about how the animal is interacting with the environment or what kind of stimuli were presented, and behavior recordings and so on. And the very first thing that we do, actually, I think this relates to the question that was raised earlier, is to consolidate all of these data streams into a single NWB file. And the reason why we do this is because, one, we want to share the data immediately. And NWB, as we know, is is a nice format for doing that, since as long as you know the API, anyone can access it. And also, we're going to be doing analysis on the NWB file. And because we'll be sharing then the data in the NWB format, we want the analysis to apply to the NWB file rather than the raw data. That way, anyone who has our analysis code can actually reproduce every step in the analysis. And this is done by a package that we have been developing with a company called Rector-NWB, so-called because we use hardware from a company called Spike Gadgets, and they save their raw data in .rack files. And this is all, by the way, this is available from our GitHub. So if you're interested in checking it out, feel free to do so. And we have organized our NWB files such that each NWB file corresponds to a single data recording from a single animal. So typically in a project, you may have multiple of these NWB files. And the next step is to insert the information that is present in the NWB files into the data joint database. So as I said, this is like a relational database for scientific data. Here's a sort of an overview of the architecture of our data joint database, which consists of, I know you can't really see these things very well, but I just wanted to point out that each of these blobs, circles and rectangles, correspond to a single table, and they have hierarchical organization. So some of these are upstream of others. And like a nice feature is that if you delete something from an upstream table, then all the associated entries in the downstream tables also get deleted together. That way, the integrity of the data is maintained and there are no orphaned entries. And the architecture largely mirrors that of the NWB files themselves. So for every group and data sets within the NWB file, we have a corresponding table in the data joint database, roughly speaking. And although it is possible to store the NWB file directly in the data joint database, we have actually decided to store only the metadata and the analysis parameters, and actually just have pointers to the raw data in NWB files for the cases where we need to access the raw data. And we found that to be an efficient way of retrieving data. And because we work in Python, so we use the Python version of data joints and PyNWB as well, we also have access to other analysis tools. For example, for doing LFP analysis, we use a package called Ghostify, developed by Joshua Chu, which efficiently does things like spectral decomposition and wavelet transformation. And for spiking data, we use Spike Interface, whose development has been led by Alessio Bacino. And this is a very nice tool that allows us to manipulate electrical recordings in the NWB file as objects in Python. So we can apply different pre-processing steps, call different spike sorting algorithms, and compute quality metrics to assess the results of spike sorting. And this is just a more of an in-depth view at the spike sorting part of our data joints architecture. So we have a table called session where basically this is contains an entry. Each entry corresponds to an NWB file. And from the NWB file, we automatically populate some of the downstream tables. So for example, the sort group table contains information about the electrodes. So here, each electrode group is actually a tetrode. And it also populates the different intervals that were present in the NWB file. So we have some run sessions and some sleep sessions where the animal's navigating and then confined to a little box for resting and so on. We also have other tables like sorter, which lists the different spike sorters that are available, and the sorter params, which basically contain parameters that are associated with these individual spec sorting algorithms. And what the user would do when they want to do spec sorting is essentially just make some decisions about what they want to combine. So here the user has decided that they want to sort the second tetrode from the run one session, using mountain sort and this parameter. So they just enter this into this spec sorting params table and then call this populate method, which under the hood calls spike interface functions to run spike sorting and then save the results in a new file. So yeah, like I said, these results from some of these involve or generate intermediate results, and they're also saved in the analysis NWB file, or what we call analysis NWB file, which is basically another type of NWB file that contains these things. And we find that that's again quite nice for sharing because NWB is largely self-documenting. And after basically getting to this step, each individual lab member would write more project-specific analysis code to visualize the data and so on, and eventually reach some kind of scientific conclusions. And a recent addition to our spec sorting pipeline is Sorting View, which is a web application that easily enables the sharing and visualization of spike sorting. So with this, we can basically send our collaborator a URL, and they can just open it up in their browser wherever they are in the world, and look at the results of spike sorting and do things like apply manual curation labels and so on. Oh, and by the way, this process is also handled by NWB data join, which is a Python package that our lab has been developing, which is also available from our GitHub. So how do we share the data and the code in this framework? So let's say the project has completed and the paper is published. We would typically upload our NWB files to Dandy, as we have heard from Uli, similar to that. So, and that's nice because it provides long-term storage for these NWB files. But sometimes we may want to share the NWB files as well as the analysis NWB files with our collaborators at an earlier stage in the project. And for that, we use something called Cachery. This is another open source Python project that acts sort of like a file system manager. And one nice feature is that it allows us to easily create a decentralized network of peers, sort of like BitTorrent, if you know what that is. And as long as you have registered your NWB file in the Cachery directory, or Cachery storage, anyone in your network has access to it. So that's another way in which we can share the data. And these arrows, by the way, are bidirectional, because we can also imagine pooling some data from Dandy or receiving data from our collaborators using Cachery and then running it through our pipeline. And what about the code? So as I said, the data is shared in the form of NWB file, and we want to share every step of our analysis. So we would actually be sharing this NWB data join package, which has information about how this architecture of our data join database is constructed, and some project-specific analysis code that basically takes the user or whoever is looking at the data through the process of inserting the NWB files into the data join database, and then applying some more project-specific analysis to, for example, regenerate the figures that are contained in the publication. So I'd like to end by providing a summary and discussing a few lessons that we have learned. I think we have learned that NWB really can be effectively combined with other open source tools to create a scientific data pipeline. I know I haven't really shown you this, but I would encourage you to go to our GitHub, pull the repository and try it out. Another thing that we have learned is that during this process, we have had to make a number of decisions about how to combine these tools and so on. And one thing that we found helpful in resolving these issues is to focus on sharing, essentially. So for example, the decision to start everything with NWB file, that came from our desire to share the data immediately and also make the analysis code reproducible. So that could be an interesting design principle if you're planning to build your own pipeline. And we're excited because we think a pipeline like ours can actually enable truly data-intensive science. So you can imagine forming a hypothesis based on the data set that you collected, and then pulling a large amount of data from public repositories like Dandy, and actually testing your hypothesis on the data that has been already collected. There are a lot of problems in neuroscience experiments, such as low sample size, and we might be able to address that by combining multiple data sets in a meta-analysis style of a study. So I think that's quite an exciting perspective. And finally, although the open source projects that we have used in building our data pipeline were relatively well documented and easy to use, it still requires a considerable amount of proficiency in computational skills like Python programming. So that, I think, will continue to be sort of important for staying relevant as scientists in the future. And with that, I'd like to thank the people who were involved in this project, either directly and indirectly. Lauren Frank, who provided the vision, as well as quite a bit of code to these projects, NWB Data Joint and Recto NWB. Eric, who's also done the same, and members of Frank Lab who patiently tested this new system, even though there are a number of glitches along the way. We also like to thank the NWB team, Ryan, Chris, Ben, Oliver, and Andrew, Data Joint, Tin, Dimitri, Shen, Jeremy and Jeff from Flatiron, who developed the sorting view and cachery apps, Joshua Chu and Caleb Kamir for Ghostify, Alessio Lucino, T.S. Henning for Spike Interface. And if you're interested in looking up all the tools that I have mentioned in this presentation, here are where we can find them. So thank you, and we have to take any questions.